In this video tutorial, I'm going to introduce you to some of the key functions of the adaptive immune response. This video will comprise part 1 of our intro to the adaptive immune response and will focus on the generation of lymphocyte receptors in primary lymphoid organs and the antigens that those receptors recognize. Lymphocyte development occurs primarily in the primary lymphoid organs, and that includes the bone marrow and the thymus. The bone marrow is the site of B cell maturation. B cells that leave the bone marrow are often classified as naive B cells, meaning they are mature, but they have never seen their antigen. Some developing T cell precursors can migrate out of the bone marrow and into the thymus. And the thymus is the site of T cell maturation, and T cells that leave the thymus are mature, naive T cells ready to be activated. Within the bone marrow and the thymus, developing B and T cells undergo a process of DNA recombination to develop diverse antigen receptors. Developing B and T cells don't express a coding gene for an antigen receptor. Rather, they have to undergo a process of DNA recombination in which segments from different areas of the antigen receptor locus are recombined together, and these are called VDJ segments. And when they are recombined together, they form a coding gene that can combine with a constant region to form an antigen receptor. Lymphocyte progenitors undergo this DNA recombination to link these segments together to have a coding gene. And every T and B cell therefore undergoes a distinct process of combining different segments together to get a coding gene, meaning that every B and T cell has its own unique antigen receptor. Each antigen receptor expresses a variable region and expresses a constant region, as illustrated in this cartoon. B cells can actually change their constant region expression, and the constant region defines what we call the antibody isotype. For example, B cells that express the mu constant region are called IgM B cells. B cells that express a gamma constant region are called IgG B cells and produce IgG antibody. Autoreactive lymphocytes are eliminated in the bone marrow, and this occurs for both B cells in the bone marrow, but also occurs for T cells within the thymus. Now, the B cell receptor is comprised of two identical heavy chains combined with two identical light chains. The light chain locus and the heavy chain locus both undergo VDJ recombination. When the two chains bind together, they form an antigen binding pocket. And this pocket is often referred to as the variable region of the antibody. This region down here is often referred to as the constant region and defines the antibody isotype. Now, when I say antibody, sometimes I'll also refer to these as immunoglobulins. For the purposes of my teaching, antibody and immunoglobulin are synonymous with each other. Now, the other thing to note is that this antibody is bivalent, meaning it has two distinct antigen binding sites. They're both identical, they both bind the same antigen, but it's bivalent. The T cell receptor, on the other hand, is monovalent. It's comprised of two chains that bind together covalently, but the main difference here is that whereas B cell receptors can be either membrane bound or secreted, T cell receptors can only ever be membrane bound, and they are also only ever monovalent. Okay, so what are the antigens that are recognized by B cells and T cells? Well, B cell receptors can bind all sorts of different things. Anything that is a soluble, free-floating, or surface antigen, anything it can get access to. That can include things like soluble proteins. It can include things like nucleic acids and polysaccharides and carbohydrates, and the surfaces of pathogens as well. And so B cells are very flexible in the types of antigens that can activate them. T cells are much less flexible. T cells can only be activated by peptides. These are strings of amino acids derived from pathogens that are 8 to 20 amino acids long, and they're presented or bound to this complex of molecules called the major histocompatibility complex. This major histocompatibility complex presents these peptides to T cells leading to their activation. So as I mentioned, activated B cells can also secrete their receptor. So a naive B cell, seen here, has only surface expression of its B cell receptor. It does not secrete antibodies or immunoglobulins. In naive B cells, this surface expression is usually IgM or IgD isotype. But activated B cells start secreting antibodies. They can secrete immunoglobulins, and initially activated B cells only secrete IgM. And I brought just a little picture of the cartoon I showed you a minute ago to illustrate that. You can see that mu, the constant region that encodes the IgM constant region, is the first to be expressed. But this locus can undergo further DNA recombination and change its constant region, and it can switch to start secreting a different antibody isotype. So activated B cells can start out secreting IgM, but can switch to become an IgG or IgA or IgE producer. A single B cell only secretes a single isotype at a time, but that B cell can switch. Okay. T cells, like I mentioned, are activated by peptide antigens. These are antigens, amino acids, strings of amino acids that are bound to MHC molecules. The MHC molecule itself is membrane bound, and there are two classes of MHC, MHC class 1 and MHC class 2. MHC class 1 is expressed by all nucleated cells, whereas MHC class 2 is only expressed by a class of cells called antigen-presenting cells, which includes dendritic cells, macrophages, and B cells. So here's just another picture of that T cell that has a T cell receptor that is binding to this small peptide being presented by this major histocompatibility complex molecule. There are two classes of major histocompatibility complex, MHC class 1 and MHC class 2. And these two classes activate the two major subsets of T cells, the CD4 positive T cell and the CD8 positive T cell. CD4 positive T cells are activated by MHC class 2. And this is because CD4 is expressed on the surface of these CD4 positive T cells and stabilizes the binding of this T cell receptor to MHC class 2 molecules. Conversely, CD8 positive T cells are activated by MHC class 1. That is because CD8 is expressed on the surface of this subset of T cells and binds to MHC class 1 molecules and stabilizes the binding of the T cell receptor to that complex. Okay, so in humans, when we say major histocompatibility complex, you'll often hear the term human leukocyte antigen, or HLA. MHC is equal to HLA in humans. Okay, so when you hear HLA, think major histocompatibility complex. Major histocompatibility complex genes are on the same chromosome. They're tightly linked together. There's 3 MHC class 1 genes, HLA A, B, and C, and there's 3 MHC class 2 genes, HLA DP, DQ, and DR. Now, if we look at the binding of a peptide, here illustrated when we look at an MHC molecule top down, we see the binding of this peptide with this MHC molecule. It's important to note that each MHC molecule can bind a range of peptides based on their biochemical characteristics. And this binding is non-covalent but stable and relies on hydrogen bonds and ionic bonds, but not on covalent bonds. Under non-infectious conditions, MHC molecules can bind self-peptides, but these don't activate T cells. But under infectious conditions, MHC presents foreign peptides derived from pathogens to T cells, and this leads to activation of the T cell response. Now, we have 6 unique MHC genes, 3 MHC class 1 genes and 3 MHC class 2 genes, as I mentioned to you in the last slide. We also have 2 alleles of each of these genes, so this increases the diversity of peptides that can be presented within an individual. But within communities, MHC is also highly polymorphic. In fact, the MHC locus is the most highly polymorphic locus within our genome. And there are thousands of polymorphic variations of each MHC gene. And so this increases the diversity of peptides that can be presented within a community or within a population. Okay. all nucleated cells express MHC class 1. And this just gives you an idea of what that looks like. There's two HLA-A genes, two HLA-B genes, and two HLA-C genes. And the specific polymorphism that is expressed is often indicated by a number, so HLA-C12 or HLA-A2. And every human has their own set of HLA molecules, their own panel of HLA molecules that they express. Professional antigen presenting cells like dendritic cells, macrophages, and B cells also express MHC class 2. And so here's an example of a macrophage that has all of the MHC class 1 genes, but now we've added in all of the MHC class 2 genes as well. So this expression is codominant. Okay. That is the end of this lecture. Sorry, I went briefly into the intro to the next one, but that's the end of this tutorial. And so this is will set us up for understanding now how B cells and T cells get activated. And that will be part of the next tutorial.